Welcome back, guys, to my YouTube channel. My name is Patrick Webster, and I will be teaching y'all about biology today. So today in biology, we will be learning about the KRAS mutation. You ready to go on the ride? I am about to show y'all my 3D model in an illustrated version of the mutation. Version of the KRAS mutation. So we have the wild type, and then we have the mutant KRAS type. This is my 3D model of my KRAS mutation, and I'm gonna show y'all which one is which. So as you can see, this is the wild type KRAS mutation. Yep. And then over here on the other side, we have the mutant type as it goes through the mutation, as it turns from the wild type to the mutant type. Yep. And in the wild type KRAS, you can tell that there are more protein linked to more proteins. Then when you get to the mutant, there are less proteins that are connected to each other. So as you can see in the wild type, the pathways, like the signal pathways, they're more just one pathway instead of multiple, as you can see in the mutant type. They're more all over the place, more inflamed, and they turn red. All right, welcome back. Now I am about to answer all the questions and concerns about my 3D model, the KRAS mutation. So it is mutated in about 10 to 30% of cancer. So what is the normal function of this protein? Explain why you chose this category. The KRAS gene serves as a central regulator of signal pathways responsible for cell proliferation, differentiation, and survival. Basically, in other words, in like condensed words, it's cell division. So I would consider the KRAS as a relay molecule because it sends signals around the cell. So then we're going to go into the next question. Where is this protein normally found? So all human cells contain the KRAS gene. And then number 3, we're going to categorize the type of change the mutation has on the protein function. Explain why you chose this statement in relation to how the mutation works. So I chose that the protein becomes more active. Because when mutated, the KRAS gene has the potential to cause normal cells to become cancerous. So it gets more active. It turns everything. So yeah. So then the next question was, how does the change in structure impact the mutated protein's function? I said it changes the protein's form, causing it to change the protein's function. So when the form changes, the protein's function changes. Yeah. Just like if we change, the body gonna change. You feel me? So yeah. All right. So now we're about to describe the KRAS gene and like name the functional groups and stuff of the original amino acid and the mutant amino acid. So we go from glycine to cysteine. So that's the original amino acid, which is glycine, and the mutant amino acid is cysteine. So glycine has 2 functional groups, the carboxyl group and the amine group. And the original amino acid side chain are capable of creating hydrogen bonds because it has that side chain of hydrogen atoms. So because of its minimum side of hydrogen atoms, glycine can be both hydrophilic and hydrophobic. Compared to other amino acids, glycine is also the smallest and simplest naturally occurring amino acid. All right. And the type of bonds it can make is hydrogen bonds. And that's because of its side chain of hydrogen atoms. And then we're going to go on to the cysteine and the functional groups of cysteine. So the amino group and the carboxyl group and the sulfonyl group are part of the cysteine. And cysteine mutated amino acid side chain is considered polar and hydrophobic. Cysteine is also just like glycine, the smallest naturally occurring amino acid. If you look at their structure, they're about the same size. And in comparison to the original amino acid side chain that has hydrogen bonds that make it hydrophilic to water, the mutated amino acid side chain has a disulfide bond, which makes it capable of stabilizing proteins and promoting stability. That's pretty cool. You feel me? So yeah. In the KRAS protein, the mutation alters single protein building blocks. The altered protein shows an increase in GTP binding levels and decrease in GTP to GDP conversion capacity. Such as effects can result in sustained activation of the protein KRAS. And some of the limitations that I got was that about my 3D model is missing details. We're only showing the glycine and the cysteine instead of showing all the other proteins that could be part of the KRAS gene. We're not really seeing the full change of the amino acid. We're just getting a visual representation. We learned theoretically, but still can't see the whole change of the amino acid. You understand what I'm saying? So yeah, that's the KRAS gene. Hope you learned something.